or from fat. They split into hydrogen ions and electrons. The hydrogen ions are then actively pumped. They are actively pumped, or the protons are pumped by a proton pump. In other words, using energy from the matrix into the intermembranous space. That is between the inner and outer membranes of the mitochondrion. Where the hydrogen ions accumulate, become too many, their concentration in the intermembrane space becomes higher than that in the matrix. That is why the intermembrane space has a lower pH than the matrix. But when they accumulate, the inner mitochondrial membrane becomes impermeable to protons. Therefore, they can't move back to the matrix. However, on the cristae, there are channels which we are calling the Chemiosmotic Channels. And as such, the protons now diffuse from the intermembranous space across Chemismotic channels back to the matrix of the mitochondrion. But in the process, as they diffuse across the Chemiosmotic channels, it leads to formation of ATP by combination of ADP with an inorganic phosphate. ADP combines with an inorganic phosphate, forming ATP in presence of ATPase enzyme. ATPase enzyme. When the protons reach the matrix of the mitochondrion, they recombine with the electrons, forming hydrogen atoms that combine with oxygen, leading to formation of water. So that is the chemiosmotic synthesis of ATP, showing ATP being formed within the chemiosmotic channels as a result of protons diffusing from the intermembrane space back to the matrix of the mitochondrion. As they leave the matrix, actively pumped, proton pump. As they move back to the matrix across chemiosmotic channels, they simply diffuse. So that is how energy is synthesized in the or by chemiosmosis. By chemiosmosis. Now, what are the adaptations of the mitochondrion for chemiosmosis? Because we have seen, we have noted that chemiosmosis totally occurs within the mitochondrion. Protons moving from the matrix to the intermembrane space, then from the intermembrane space across chemiosmotic channels back to the matrix. Therefore, what are the adaptations of the mitochondrion for that? When we said the mitochondrion has stocked particles called the oxysomes with pores that allow hydrogen ions from the intermembranous space to the matrix. Those channels or those pores are the channels which we've called the chemiosmotic channels. Then we've said that these stocked particles, also the oxysomes, also contain ATPase enzyme, which ATPase enzyme is needed for synthesis of ATP from ADP and an inorganic phosphate. Remember that the inner membrane is highly folded into cristae, and that increases the surface area for chemiosmosis to take place. Then the inner mitochondrial membrane is thin for quicker movement of ions across it. So those are the adaptations of the mitochondrion for that process of chemiosmosis. We then look at ATP and energy yields. Some calculations here, but simple calculations, very easy calculations. Now we have noted that respiration occurs in 3 stages: glycolysis, Krebs cycle, ETS. Where in ETS, it is basically formation of ATP from the hydrogen atoms that are transferred by NAD or fat. Let us see how many ATP molecules are liberated from a single glucose molecule. Here we are moving step by step so that we come to the total number of ATP molecules liberated. During glycolysis, we saw that from one molecule of glucose, we end up forming 2 molecules of pyruvate. But in the whole process of glycolysis, there were 2 ATP molecules liberated from each glycerate 3-phosphate. But because, look at the point where we have fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate splits into 2 molecules of glycerate 3-phosphate. It splits into 2 molecules of glycerate 3-phosphate, also called triose phosphate or TP. But each glycerate 3-phosphate liberates 2 ATP molecules directly. Meaning, since there are 2 molecules, that means how many ATP molecules are liberated from one glucose molecule directly in glycolysis? Those are 4 ATP molecules. Why 4? Because look at the right-hand side, your right-hand side, the right-hand side of that cycle. From glycerate 3-phosphate to pyruvate, we have 2 ATP molecules liberated directly. But because there are 2 glycerate 3 molecules leading to formation of 2 pyruvate, how many ATP molecules are liberated directly? That gives us a total of 4 ATP molecules liberated directly. How about indirectly? We get back. Indirectly, we are looking at the ATP molecules that are to be liberated from NADH2 when it reaches the electron transport system. When we look at glycolysis here, how many NADH2 molecules are we liberating? From one molecule of glucose, we form 2 pyruvate. But before forming pyruvate, glycerate 3-phosphate to pyruvate, how many NADH2s are liberated? 1. But because there are 2 on either side, on the right and on the left, because there are 2 glycerate 3-phosphates, that is why we say we liberate 2 NADH2, 1 on either side. Now when we liberate 2 NADH2, each NADH2 liberates or generates 3 ATP molecules like we've seen from the ETS, giving us a total of 6 ATP molecules indirectly, meaning 4 ATP molecules directly and 6 ATP molecules indirectly. That is from the NADH2 for each molecule of glucose. And as such, when we add the 4 directly to 6 indirectly, we end up with a total of 10 ATP molecules. 10 ATP molecules. But remember that there was use of ATP, glucose to glucose 6-phosphate, fructose 6-phosphate to fructose 1,6-bisphosphate. ATP molecules were used. Therefore, how many ATP molecules were used in the whole glycolysis? They are 2. Meaning, if we have formed the 10 ATP less the 2 which were used, we remain with a net yield of 8 ATP molecules. Meaning, for glycolysis to form ATP, we need to use ATP. ATP is used to liberate ATP. That's why from glycolysis we are having a total of 8 ATP molecules as the net yield. Now, after glycolysis, pyruvate, before the pyruvate goes into the Krebs cycle, remember pyruvate is decarboxylated and dehydrogenated. When it loses the hydrogen atoms, they combine with NAD. forming NADH2, which NADH2 is taken to the ETS. But how many pyruvate molecules do we have? From glycolysis, they are 2. Meaning, how many NADH2 molecules do we have? They are also 2. That is why we are saying that before the Krebs cycle, there are 2 NADH2 molecules. And each NADH2 liberates 3, giving a total of 6 ATP molecules. Meaning, glycolysis, a total net of 8. Before the Krebs cycle, from the pyruvate, a total of 6 ATP molecules. Then, how about when it comes to the Krebs cycle? Look at that cycle, look at the Krebs cycle, look at that cycle. Look at the points where ATP is liberated directly. We have the conversion of alpha-ketoglutarate to succinate. Alpha-ketoglutarate to succinate will liberate 1 ATP directly. But because we had 2 pyruvates, that means in total each gives 1 ATP directly. Hence, we are having 2 ATP molecules liberated directly from the Krebs cycle. 2 ATP molecules liberated directly from the Krebs cycle. Because we are seeing alpha-ketoglutarate to succinate 1 ATP. But because there are 2 pyruvates, that's why we are multiplying it by 2. Then, how many NADH2 are liberated in the Krebs cycle? Look at it. Look at it. Count. Count. We are seeing the first NADH2 citrate to alpha-ketoglutarate, that is 1. Alpha-ketoglutarate to succinate, those are 2. Malate to oxaloacetate, those are 3. NADH2. But because there are 2 pyruvates, we multiply each by 2. Therefore, 3 times 2 giving us a total of 6 NADH2. 6 NADH2. But because each NADH2 liberates 3 ATP molecules, that is 6 NADH2 times 3, giving a total of 18 ATP molecules liberated indirectly from each NADH2 in the Krebs cycle. How about the third H2? Look at it. Only from succinate to malate is where we have third H2. It is 1 multiplied by 2 because of the second pyruvate. We end up with how many third H2? We end up with 2 third H2. But each third H2 liberates 2 ATP molecules from the ETS, giving a total of 4 ATP molecules. Therefore, glycolysis, a net yield 8 ATP. Before Krebs cycle, a net yield 6 ATP. Krebs cycle gives us 2 ATP molecules directly, 18 indirectly from NADH2, and 4 indirectly from FADH2. Therefore, 8 plus 6. 8 plus 6. Simple mathematics. 8 plus 6, that gives us 14. Plus 2, 16. Plus 18, 34. Plus 4, 38. That is why we get an overall yield of 38 ATP molecules from 1 molecule of glucose. Therefore, from a single glucose molecule, we end up yielding a total of 38 ATP molecules. 38 ATP molecules. Allow me to stop there for today. It's been nice going through this lesson. I really think that you've understood something out of it. When we meet again, We shall now look at the last part of respiration, and that will take us to what we call the phosphocreatine system. You can read through the phosphocreatine system so that when we meet again, at least you have some idea about it. Remember the assignment that we've given, comparing oxidative phosphorylation with photophosphorylation. Keep safe, stay well. Until we meet again, have